Hello friends, today we will learn about Hemophilus and Gardnerella. Here in this video we'll study about the Hemophilus influenza's morphology, culture, biochemical reactions, antigenic structure variation, sensitivity to physical and chemical agents, virulence factors, pathogenesis, laboratory diagnosis, treatment, prophylaxis, and then we'll study about some Hemophilus, other Hemophilus. Under this, we'll study about Hemophilus aegyptius, Hemophilus ducreyi, Hemophilus parainfluenza, Hemophilus hemolyticus, Hemophilus arthropophilus. Then we'll study about Hasek group bacteria. Then we'll study about Gardnerella vaginalis, its morphology, culture, biochemical reactions, pathogenesis, clinical features, diagnosis and treatment. So, let's begin. Genus Hemophilus contains non-motile, non-sporing, gram-negative bacilli and require one or both of two accessory growth factors, that is X and V, present in blood. Hemophilus meaning blood-loving. A scientist named Pfeffer in 1892 isolated the organism from the sputa of patients from the influenza pandemic and proposed this as the causative agent of human influenza. This came to be known as influenza bacillus, Pfeffer's bacillus. It was renamed as H. influenza. The discovery of influenza virus in 1933 established that H. influenza was only a secondary invader. H. influenza and H. ducreyi are the major pathogens in the genus Hemophilus. H. influenza may cause meningitis, pneumonia, epiglottitis, bronchitis, otitis media, septic arthritis, and H. ducreyi is the causative organism as cancroid. Now let's study about Hemophilus influenzae. Let's study about the morphology. It is a small 1.5 microgram into 0.3 micrometer gram negative non-motile bacillus showing considerable pleomorphism. It is non-sporing and non-acid fast. In long culture 18 to 24 hours, the cells are usually cocobacillary, while in older cultures, long filamentous forms may be seen. In sputum, they occur as small clumps of cocobacilli and in CSF from meningitis cases. Long and filamentous forms predominate and virulent strains possess capsule while the avirulent strains and old cultures are non-capsulated. Now let's study about the culture. H. influenza has fastidious growth requirements. It grows better in aerobic than in anaerobic conditions. It requires enriched media such as blood agar or chocolate agar because the accessory growth factors known as X and V present in blood are essential for growth. The optimum temperature for growth is 35 to 37 degrees centigrade. Some strains require 5 to 10% carbon dioxide. It cannot grow on nutrient agar which lacks the accessory growth factors. Now let's study about X factor. It is a heat stable protoporphyrin IX hemine or other iron containing porphyrin. It is necessary for the synthesis of catalase and other enzymes. Cytochrome C, cytochrome oxidase involved in aerobic respiration. Now let's study about V factor. It is a heat labile factor present in the red blood cells and in many other animal and plant cells. It is synthesized by some fungi and bacteria. It is either nicotinamide adenine dinucleotide, in short NAD, coenzyme 1, or NAD phosphate coenzyme 2. That means, in short, NADP coenzyme 2. It appears to act as a hydrogen acceptor in the metabolism of the cell. Ordinary blood agar is not suitable for the growth of H. influenza, where growth is scanty as the V factor is not freely available. Being imprisoned inside the red blood cells, B factor is released from erythrocytes in chocolate agar, that is heated blood agar, at 80 to 90 degrees centigrade. V factor is also synthesized by Staphylococcus. Growth on blood agar can be improved by providing a source of V factor, Staphylococcus aureus. The differential requirement of X and V factors helps to distinguish various species of Hemophilus. Blood agar with Staphylococcus aureus streak and chocolate agar are routinely used for the identification of H. influenza. The colonies are small, translucent, and non-hemolytic on blood agar. Capsulated strains produce distinctive iridescent colonies. Better growth of H. influenza is also obtained on Levinthal's medium or fields agar by adding a peptide digest of blood in nutrient agar. Now let's study about satellitism. Although blood agar contains X and V factors, colonies of H. influenza are small due to non-availability of V factor. After inoculating suscepted H. influenza on a blood agar plate, Staphylococcus aureus is streaked across the same blood agar plate and incubated at 37 degrees centigrade for 18 to 24 hours. The colonies of H. influenza will be large and well developed alongside the streak of Staphylococci, while those further away from staphylococcal streak are smaller. This phenomenon is called satellitism and demonstrates that V factor is available in high concentration near the staphylococcal growth and only in small quantities away from it. Now let's study about biochemical reactions. H. influenza is catalase and oxidase positive, ferments glucose and galactose, reduces nitrate to nitrite. It does not ferment sucrose, lactose, and mannitol. On the basis of production of indole urease, ornithine decarboxylase is decarboxylase. It is divided into 8 biotypes. That means 1 to 8. Majority of clinical isolates belong to biotype 1 and 3, and type B organisms belong to biotype 1. However, biochemical reactions are not helpful in identification. Now let's study about antigenic structure. Isolates occur in capsulated and non-capsulated forms. Three major surface antigens are present: capsular polysaccharide, outer membrane protein, and lipo-oligosaccharide. The capsulated strains produce a capsule which is polysaccharide in nature. On the basis of capsular material, Pittman divided H. influenza into 6 serotypes: A, B, C, D, E, and F. Capsular serotype B strains are associated with most invasive infections. Serotyping of H. influenza was originally done by agglutination reaction using type-specific antisera. Other methods include Quelling reaction, coagulation, CIEP, and ELISA. Diagnostic kits for the identification of H. influenza type B are available. The type B capsular polysaccharide has a unique chemical structure containing polyribosyl ribitol phosphate, which induces IgG, IgM, IgA antibodies. These antibodies are bactericidal, opsonic, and protective. HIV HIBPRP is therefore employed for immunization. Non-capsulated strains cannot be typed and are called non-typable strains. OMP antigens of HIV has been classified into 13 subtypes. LOS are antigenically complex. OMP and LOS subtyping are of epidemiological value. The complete genome of H influenza has been sequenced. Now let's know about the variation. Colonies of H influenza show a smooth to rough variation accompanied by loss of capsule and virulence. Genetic transformation has been demonstrated in H influenza to transfer the characters of capsular antigens and antibiotic resistance. Non-capsulated strains become capsulated by genetic transformation. Now let's study about sensitivity to physical and chemical agents. It is a delicate organism. It is readily killed by heat that is 55 degrees centigrade for 30 minutes, refrigeration at 4 degrees centigrade. Disinfectants and drying cultures may be preserved on the chocolate agar. For long term preservation, the culture may be lyophilized. Now let's study about virulence factors. Capsular polysaccharide, it resists phagocytosis. Loss of capsule leads to loss of virulence. Let's know about the pili, fimbriae. They help in attachment of organisms to epithelial cells. Now, let's study about outer membrane proteins. They contribute in adhesion and invasion of host tissues. Next point is IgA1 protease. H. influenza produces a protease that specifically cleaves the heavy chain of IgA1. Now let's study about pathogenesis. It is an obligate human parasite